We're here in front of the iconic Opera House in beautiful Sydney, Australia, to learn about Nanosonics, a global leader in innovative infection prevention solutions. Founded on the principles of innovation and dedication to patient safety, Nanosonics has emerged as a global leader in automated high-level disinfection technology for ultrasound probes. From their cutting-edge trophon 2 device to their advancements in ultrasound probe compatibility, Nanosonics continues to raise the standard in infection prevention. Join us as we take you behind the curtain, exploring the minds behind the technology and the lengths they go to ensure safe use of ultrasound probes on this episode of Beyond the Tour. Before I left for Sydney, I wanted to prepare by talking with a big player in the ultrasound probe space. So I connected with Reg Rumwell, resident expert at Siemens, to pick his brain before my trip. Reg, thanks so much for jumping on a call with me today. Me and my colleagues here at Beyond Clean are heading out to Australia, and I would love to hear from you from the manufacturer's perspective. Why is probe compatibility important? Why does it matter?
Sure, Linz. The reason we go to such great means in working with our partners to establish probe compatibility is primarily so that the equipment continues to function as the customer expects it to function. We provide very detailed instructions. Their instructions for use are on our website. And by following those, it really saves the customer a lot of angst in terms of having to rescan patients if they've damaged a transducer, having to get replacement equipment if they've damaged a transducer. The compatibility aspect is key. And it's important that, the customers realize the length that we go to ensure they're going to continually have a good experience with our equipment and really do the job that it needs to do.
What I'm hearing from you is that non-compatible reprocessing of your ultrasound probes and transducers can lead to ineffective equipment, images that are not clear. Is that correct?
That's absolutely spot on, Lindsay. And that costs both us and our customers an awful lot of capital as well. The reason we do all of this is such that, the hospitals can be sure that they're not having issues with HAIs, nosocomial infections. The previous topics we discussed about damage and cost of the equipment, about diagnostic accuracy of the images they're generating from our ultrasound probes. It's really super important that the customers really follow the instruction we give them and have the confidence that, we've done the homework. We've worked with our partners. We are confident when we recommend, here's how you clean or disinfect your equipment.
This has been a really great discussion. I'm excited that I was able to connect with you before going and meeting with the Nanosonics team in Sydney. Thank you so much for your time today. I really appreciate it.
You're absolutely welcome, Linz. Enjoy your journey and give my best to our partners down in Australia.
After 20 hours of travel and a few much-needed naps, Bobby and I arrived at the Nanosonics headquarters to meet up with our friend Sunshine Macaro, Vice President of Marketing for the Americas, so she could introduce us to the team in Australia.
Linz, Bobby, we are beyond excited to have you guys here. Ever since we first started talking about probe compatibility, probe damage, all of that, we've just been waiting for the day when we could welcome you here to our Sydney headquarters. We have the best of the best lined up for you today. We've got John, our Chief Medical and Scientific Officer. We've got Matt, our Strategic Partners Manager. They are gonna take you behind the scenes. But first, I gotta introduce you to our most important person, VIP, our CEO, Michael Kavanagh.
Well, thanks very much for the introduction, Sunshine, and welcome to Australia.
Thank you.
Nanosonics was formed probably just over 15 years ago with the sole purpose. We were looking at the need to transform medical device reprocessing to deliver improved patient safety and better healthcare outcomes. Our trophon technology really pioneered the automation of ultrasound disinfection. Today, there's approximately 35,000 devices in use internationally. And that's one statistic about the number of devices, but really how we think about it as an organization is that 35,000 devices really represents about 100,000 patients every day being protected against the risk of cross-contamination because the ultrasound transducer that is used on that patient has been decontaminated in the trophon device. So when you think of 100,000 patients every day, but that equates to over 30 million patients annually. So we're very, very proud of that fact. So over the course of our time together, I think you're going to get a great insight into the details involved in the probe compatibility program, not just what we do, but what all the manufacturers of the ultrasound products do as well. So welcome.
Thank you.
Thank you.
We're very excited, not only to be in Australia, which is awesome, but also to look at this kind of niche of medical devices that, at least in North America, feels like hasn't had a spotlight shined on it in a very long time. So really looking forward to it. Thanks for having us.
You're welcome.
From here, we headed out with John and Matt for a short walk from the Nanosonics headquarters to the production facilities to begin the tour.
So here we are outside of our lab and manufacturing facility. So just behind me is where we do the manufacturing of trophon, and these are our labs where we do all the testing on the device. Trophon does high-level disinfection of ultrasound probes. So an ultrasound probe obviously is a reusable medical device in most cases. They're quite expensive, and so we have to make sure that the microorganisms from the first patient are not transmitted to the second patient. And that involves cleaning and high-level disinfecting the probe. The way that trophon works is it actually uses hydrogen peroxide. It nebulizes this into a fine mist, and that fine mist coats the probe and disinfects it within the chamber. Okay, so let's go inside and look at some of the labs.
Awesome.
Let's go.
All right, so Linz, Bobby, I'm really excited to finally show you our compatibility labs. And what probe compatibility is really safety for patients, safety for users, safety for equipment. We'll start with Bioscience lab, where we make sure that we kill all the pathogens. We'll go to the chemistry lab to make sure that the probes are safe to handle for our users, and then go to our material labs, where we look at safety for users and equipment.
Let's
Sounds
do it.
good. Yep.
I wanted to introduce you to Kieran, who runs the Biosciences lab.
This lab is focused on the trophon efficacy testing or any OEM testing. We do the production quality control as well. So new batch of trophons being built. So we run the monthly quality control testing as well in this lab.
So you get to play with pathogens all day is what you're saying.
Yes.
So, okay, great. How does that process work? Are you using real pathogens in here, or is it substitute test soils? How do you do that?
That's also a good question. So what the process is, like you can see in the background, the real probes, they are actually contaminated, inoculated at multiple locations using a real pathogen.
Okay.
Probes are then reprocessed through the trophon. And after the reprocessing, take the probes back and try to extract what is the residual bioburden. So we ensure that there is, as per the regulatory requirements, there needs to be achieving 6 log reduction in the bioburden on the total probe area to achieve that high-level disinfection. So it's not only one microorganism we're working on. During the development of the device, we tested a broad range of microorganisms, bacteria, fungi, viruses, spores, everything, so that we ensure that trophon can be successfully, efficiently used to high-level disinfect the probes before and after patient use.
So you have to keep all those different pathogens in these refrigerators around the room. That sounds terrifying.
No storing your lunch in these ones.
No food to be stored.
You mentioned outside that hydrogen peroxide is the method that we're getting that high-level disinfection. I'm curious, why hydrogen peroxide as your disinfection modality?
Yeah, great question. So it's an oxidative chemistry. So it's really hard for microorganisms to evolve resistance to this because it has a very general mechanism of action, unlike some antibiotics. So you really don't see emergence of resistance to peroxide in a meaningful way for the pathogens that we deal with in clinical use. There's over 1,300 probes that are compatible with trophon. They're all different shapes and sizes, and a lot of them have special features like indentations where biopsy guides are clamped onto the probe. So we take a lot of care actually in ensuring that every single probe is disinfected effectively with trophon, high-level disinfected, by doing all of that specific testing on all of the individual probe models.
We're starting in this lab, and already the depth and thoroughness that you're describing is sort of setting the tone for this tour. I am so excited for what's to come.
After the Biosciences lab, we went to the chemistry lab to meet Bahar, the research and product development team lead.
So Lynn, Bobby, I wanted to introduce you to Bahar. She's our expert in probe compatibility. She looks after our material and chemistry lab, and that's really essential in terms of probe compatibility for us. For chemistry, we need to make sure that any level of residuals on the probe is under the safety limit, so the probe's safe for the user and the patient.
Welcome to the lab. First of all, we run a standard cycle on a probe. We use minimal chemistry. It's a couple of grams only in each cycle, and then the device itself runs the exhaust process to take out all the mist from the system before the doors are open. And then exhaust product will go to a catalytic reaction to break down hydrogen peroxide to water and oxygen. And then we take the probe immediately inside DI water. We extract the residual left on the probes using the water bath, and then we take that extract to another of our labs to analyze and calculate the amount of residual.
So essentially it's like a rinse bath, and then you're testing the water to see if any of the chemistry showed up in the water.
So it's safe, but we make sure that it's safe on every individual probe.
A critical step in the Nanosonics probe compatibility program is making sure the trophon 2 does not damage ultrasound probes. So we went to the materials lab to see their process for ourselves.
This is where we test the probe for its life cycle, making sure that from day 0 to the end of the life of the probe, the technology is safe for it. Firstly, we measure its dimensions to make sure that it fits into the trophon envelope, and then we do a fit test. We hang the probe inside the device, making sure that it fits properly. And the next step is to check the temperature of the probe surface during the cycle. What we do is to go with the worst case scenario. So we run 2 back-to-back cycles to make sure that if this is what happens in the fields, like very quick reprocessing, it doesn't exceed the limit.
Okay, so you're taking into consideration the fact that a probe can be reprocessed over and over and over again in a single day.
Yeah.
And you want to make sure it doesn't melt, essentially.
Yes.
You're not only testing the chamber temperature, which I'm sure the machine does, but you're actually putting temperature loggers and data loggers on the probe itself and measuring that.
Yeah, the important areas of the probe, which is the length of the probe and also on the body of the probe.
Okay.
After that, we check if the probe is compatible with the technology during its lifetime, from day 0 to the number of years that a probe should be in the field. We run back-to-back cycles, 24 hours a day.
So are we talking 20 cycles? Are we talking 200?
We're talking about thousands of cycles, and it could be as high as 18,000.
Wow, okay.
That probably takes a while to get through.
Yeah, a few months for each probe to go through. Every 1,000 cycle, we do photographs from certain angles of the probe that are set in our protocol.
Do you ever do usability testing to make sure that it's functioning properly?
We note down all the observations. If there is anything significant, we will communicate with the probe manufacturer. And at the end of the cycles, we send the probe back to the manufacturer, and they run the fit and functional test there.
Oh, so it's the manufacturer that gives the final say of whether or not it's still in original condition, still working the way it's supposed to. What do you do in this room to help emulate like the real world use of a probe that's actually going to be handled in a facility?
Yeah.
The main question is that a fake in there?
That's right.
Trophon step is one step of the reprocessing. The probes get wiped first, and then they get dry wiped, and then they will go through trophon, and then after that, storage and handling will be involved. We tested the probes like when, as you said, the user is not having a good day, so they push it hardly.
Yeah.
All these might affect the life of the probe.
So you're using real probes from real probe manufacturers, real trophon machines, and essentially roughing them up a little bit along the way just to account for that real world handling.
Exactly.
had an opportunity to sit down and talk with Reg at Siemens about device compatibility and how important that is. At what point in the process of manufacturing and designing a new probe do you actually get involved?
The beauty about our collaboration with probe manufacturers is that we are so involved early on that even when the manufacturers are designing the probe, they're thinking about compatibility, so that probes are designed with trophon compatibility in mind.
I can show you some examples of what can happen to a cable through just wiping.
That's just from wiping?
Just from wiping.
How would you like that to be your job, to be
Right.
the probe wiper?
And you can see on the cable difference in where it was wiped
Oh, yeah.
against where it wasn't.
Oh, wow.
And you can quite clearly see the chemistry and compatibility. So thank you so much, Baha. Now it's a good time for us to switch gears and go to the production lab, show you how probe compatibility is baked into the production of every single trophon device that's released globally. We made it to our final stop, the production area, and we've got Anastasia to help us to run you through what happens here and how we actually build trophon devices globally.
Yep, so welcome to where the magic happens. We pride ourselves on, one, being an Australian manufacturer, and two, our consistency with how we manufacture our devices. So we have in-process quality checks, so our sub-assemblies are tested on our sub-assembly line through jigs that make sure that they're functioning correctly. And then our main quality check is our end-of-line testing. So they go through a 5-hour test to make sure that our device is completely functional, so that it's ready to test a whole range of probes, and so that it's also safe for patient use.
Bobby, I can't help but think of the assembly area in sterile processing when I look at these workbenches.
Yeah, it does feel that way. As we've gone through the tour, every room I've reached for my head to feel, am I wearing my hat? And so the haircuts make me feel at home. So in my sterile processing department, we always had some music playing overhead, and people were dancing and cutting up, and part of me loved that for the culture, but it also kind of grinded against me from a quality perspective. Everybody is so focused in here, and it is super quiet. It seems like everybody takes the professionalism really seriously.
Yeah, they love their job. They love what they do. I think to know that we are actually helping patients and changing people's lives and preventing infection in the field keeps them going. Yeah. They know the value we have in our jobs.
We were just in the material lab, and what we saw there was essentially what someone in the hospital setting or in a clinic setting would see. But now that we're here in the production line area, these devices are incredibly complex.
From the front, yeah, it does look very simple, but obviously, as you can see, it is quite complex inside, and it's a very clever device in the way that it's designed to pick up anything that's detected outside its specification.
From the user experience, it's really just one button on the screen and you initiate the cycle. But as you look behind the covers at the device itself, it's actually very sophisticated.
Thank you so much, Anastasia, for your time. And I think maybe as the last thing we can do here is actually have a quick tour through the production line so you can see how trophon becomes from one single screw, one single part to the final device.
Sounds great.
Yeah. Great.
Thanks, Anastasia.
Thank you.
After the tour, Bobby had an opportunity to meet with the 3 Nanosonics regional presidents to discuss ultrasound probe reprocessing from their perspectives around the globe.
All right, guys, we just wrapped up an awesome tour of the facility, and one of the themes that kept coming up was this conversation about probe compatibility. From a North American perspective, not something that I've spent a lot of time thinking about, although it feels like there's a trophon device in every facility I end up in doing consulting. And so since I have you guys here, I want to hear what your perspective is on that probe compatibility in your respective regions.
Yeah, Bobby, in Europe, it's predominantly a manual wipe, high-level disinfection market right now. Some of them are made up of different chemistries, and some of them have limited capabilities. For that reason, it's critically important that we have a probe compatibility program. I think we have over 1,300 probes on our probe compatibility program with 25 different probe companies. So it's crucial to our market and to the other regions that we have a program in place that's passed and approved by the probe manufacturer.
The INZ market is actually quite a success story, and that is because of the efficacy and the performance of our device. There are also other automated HLD solutions which might have limited compatibility data, and so in the company we focus on assessing probe compatibility, and that is with the sole purpose of ensuring the safety of patients, the users, and also the equipment.
What people don't realize is that there's over 30,000 devices globally. And the beautiful thing about it is it's the same device whether it's in Asia, whether it's in Australia, or whether it's in the United States. It performs the same exact way, has the same probe compatibility standards that we expect of a device that's out there in the market.
I'm sure from a staff training perspective that can be really useful, but also since we're talking about patient safety and efficacy and making sure that the device is safe for use for that next patient, I'm sure that gives a lot of peace of mind as well. So guys, thanks for sharing your perspectives. Appreciate your time.
Great having you here.
Yeah.
To really go beyond the tour, we found a lovely cafe to sit down and talk about what this all means for an industry we all care deeply about.
This is my favorite part of filming these Beyond the Tour episodes because we get to come together and talk about all of the things that Bobby and I had a chance to learn. Something that's overwhelmingly striking to me is just the vastness of what probe compatibility actually means. Being able to come here and learn from your team how much you invest in making sure that the people are safe, not only the patients, but the people handling the ultrasound probes. And also that the actual probes themselves function. It's been really incredible.
It's been a huge effort. We haven't started from day one with over 1,300 probes. No, we had to start somewhere.
Right, yeah.
We couldn't just snap our fingers and here we go, they're all compatible. I remember initially it was quite a reactive approach where, we'd get a call from a rep standing at an ultrasound room with a probe asking, hey, is this compatible? And that's where it would start. Would then reach out to the probe manufacturer and would say, hey, can we test this probe? And eventually would go through the testing that you've seen, all the cycles, temperature, etc., and get it ticked. Where now the program has really evolved into much more proactive. So part of my role, which is really exciting, is working with the probe manufacturers globally and making sure that every new probe that's coming out is trophon compatible. And what's great about it now is that probe manufacturers realize that, and they work in collaboration with us.
I actually think it's really important that we have that almost full coverage of probes that are available in the market. We basically want to give healthcare professionals the choice of using trophon if they desire, particularly for semi-critical and critical procedures that require HLD. So really our mission is to have a very broad compatibility program that allows healthcare professionals to choose the probes they want to use for that particular procedure and not be constrained by compatibility.
Right, you don't want them thinking about having to pick a less desirous product because it's the one that we can get reprocessed in our machine. That makes a lot of sense. Is there anything that you all do in just partnering with your customers to try to educate around what high-level disinfection is, that Spalding classification system, and trying to encourage them to use it in an appropriate way?
Yeah, we actually have clinical application specialists in the field. They'll go and consult with end users, with sterile processing staff and so on to help educate them on what is required. So you mentioned the Spalding classification. Right. So non-critical devices only contact intact skin. As soon as the skin's broken or you go to mucous membrane contact, it gets elevated to semi-critical and you have to do high-level disinfection. And so educating people on that is really important. A certain probe might actually be exposed to different tissues throughout the day. So there might be a non-critical procedure in the morning, a semi-critical procedure at lunch, and then a critical procedure contacting sterile tissue in the afternoon.
Exactly.
So
it's the same probe. That's right. All three. Yeah. Right. And so that's what makes it so challenging with this particular issue. They think, okay, I just got some blood on this probe, so it needs to get high-level disinfected to take care of that probe. That's actually backwards thinking, right? It's too late to do the high-level disinfection after we've put it on the patient with the open wound. It really needs to be thinking, how can we get this probe to the appropriate disinfection level before we put it on that open wound in that semi-critical situation?
Right, so you don't always have prior knowledge of what the procedure is going to be like before the patient gets there, right? Emergency department is a great example. So you've got a range of different options. You can high-level disinfect that probe on demand when it's required. Now that takes a little bit of time. You've got to think about whether that can fit into your workflow. In the emergency department, you might not have time.
Right.
So a better way to do that is to have high-level disinfected probes ahead of time, right, stored in appropriate storage, bags, etc., that are basically ready to go. So you can grab those probes and use them. Another thing that I've seen facilities do is actually just high-level disinfect everything all the time, because then you're basically ready for anything.
Right, and with as easy as a process that trophon seems to have made it, it seems like the favorable solution to me. We're done with it. We just take the decision-making out of the process of trying to triage the Spalding classification, just put it in the machine after you're done with it, have it ready for the next patient. It makes a lot of sense.
Yeah, and a lot of the conversations that we have as a team at Beyond Clean revolve around this idea that the patient at the beginning of the day gets the best care versus the patient at the end of the day because of turnover, because of having to rush through steps, et cetera. And the same thing can apply here. Even running a disinfectant wipe down a probe, like everyone's going to squeeze at a different pressure. And they're going to, keep that contact on the surface for a different length of time. And there's no way to make that a safe option when what you're going for is consistent patient safety.
So all of those kind of manual considerations are taken away with trophon. It actually controls all of the critical process parameters. So in our case, it's the concentration of the chemistry, the dosage into the chamber, the temperature. The coverage is taken care of, obviously, with the mist. And then you have that chemical indicator that's a cycle verification. That's telling you the right amount of oxidative chemistry entered the chamber.
Sounds like peace of mind. Right. Yeah.
Confidence.
I've not seen any marketing materials out there of like, we can high-level disinfect most of the options, or we're the option that's going to damage your probes. So like, what would you say is like the key difference that you're proudest of to be able to make that compatibility claim and not just shout compatibility out into the open?
I think what's critical for us is the type of testing we do. So we don't just get a sample of a plastic and dip it in hydrogen peroxide and see if that seems okay or not. We do, testing with real probes, real cycles on real trophon devices, replicating that worst case, testing the temperature, testing for lifetime of the probe compatibility. And I think that's the critical part that might be missing from some other reprocessing companies. And the other key which we spoke to is that probe manufacturer approval. You really need both parties to jointly approve to confirm compatibility.
Yeah. It's impressive the work that you've put in to get over 1,300 different probes.
Kept us busy.
From a motivation standpoint, like I can't see any reason why you wouldn't be all in on making sure it's absolutely safe for devices, right?
Exactly.
But there are still, I'm sure, customers out in the field who are working with these probes. They start to see cosmetic damages or tears, and they're wondering where are these coming from. And it probably doesn't do a whole lot of good from your all's perspective just to show them a compatibility matrix and say, nah, it wasn't us. So how do you reach out to customers and help them work through those kinds of damages as they're encountering them?
What we like to do is to go in and consult with the healthcare professionals and understand, well, what are you actually doing with your probes day to day? Often we find that they're being used with an incompatible product. So they're being used with another cleaning or disinfection product that's actually not listed in the IFU for the probe. That's really common. And look, in the defense of the end users, that's really complicated because you've got all these different products that you might be using, and every IFU for every probe is potentially different. And so you've got to do this big matrix crosswalk of all these different systems, and it's very challenging. So it's handling, it's using incompatible products. Those are really probably the main causes of probe damage that we see in the field.
And as a matter of fact, when I was reviewing our post-market surveillance data, because it's a really exciting thing to look at, every single allegation that we've investigated, the root cause was never the trophon device.
Okay, that was going to be my next question.
I think people watching this might think, of course, they're the manufacturer. They're not going to find themselves at fault, right? That's easy for them to say, but I know such a fair concern. I think something else to really consider is the big probe manufacturers that we're working with. They have a vested interest as well. They don't want to be putting something in their IFUs that they're not completely confident in. They don't want to put something out that could be damaging their probes that are, high capital pieces of investment for these healthcare practitioners. So for them working with us and putting the trust in us I think that speaks volumes.
I think if you just brought every customer with a complaint to see beautiful Sydney, come see the facility, I think that would deal with
one
it.
way to do it.
Yeah, that's right.
All the months worth of work that you're doing to make sure that my probes are compatible with your machine. It just makes me feel like I can be confident. Press a button and I get to know that I'm going to have a safe probe for the patients that I'm serving.
Exactly, and that's really, again, coming back to our mission of being patient safe, but also make it easy for the users. They don't have to worry, is the probe safe? Are they doing it correctly? It's an automated enclosed system. It's signed off by the probe manufacturers and by us. So the customers and the patients can have that peace of mind.
All right, you guys, thank you so much for having us here for this tour. I know that Linz and I both came away from the tour feeling inspired, I guess is the word, which is an odd word to use. And it's because what Beyond Clean has always been about is patient safety, reprocessing instruments the right way every time, every patient, every instrument, every time. And that story has been something that I've heard in every room that we went through on the tour, with every expert that we talked to, that we're going to do the right thing for patients. We're going to do the right thing to make sure that these probes work to the best degree that they can, optimally for those patients. And yeah, I'm so appreciative that you brought us on this tour. We joked earlier about how great it could be if we could bring all of the customers of trophon to beautiful Sydney and see the facility. And I know that's not practical, but hopefully in this Beyond the Tour episode, we're able to share some of that beautiful story with the audience. So thank you so much for having us.
Oh, really? Thank you.
Yeah, for sure.
Thanks again.
Yeah, absolutely. Thanks, Lloyd.